Good morning, doctor.
Good morning.
Shall we start?
Yes, yes.
Good morning, students. As we are entering day 9 of our BIIS 12 workshop, today we are here to on the topic of application of biotechnology to veterinary medicines. To discuss this topic, we have with us Dr. Amrutlal K. Patel, Scientist B and Joint Director, Gujarat Biotechnology Research Center, Ahmedabad. He has obtained his Masters in Veterinary Science from Veterinary Research Institute, Izad Nagar, and PhD from Anand Agriculture University. He has served as a veterinary surgeon at the Vadodara Society for Prevention of Cruelty to Animals and as a veterinary officer at Baroda Dairy. He has worked at Zydus Research Center as a scientist and a research assistant at Vaccine and Infectious Disease Organization, Canada. He has also worked as a research associate at the Department of Animal Biotechnology, Anand Agriculture University. He served as a consultant for Accelera's lab and as also principal scientist at R&D Center, Hester Biosciences. He was also a research associate at the Gujarat Biotechnology Research Center and did his postdoc fellowship at Vaccine and Infectious Disease Organization, International Vaccine Center, Canada, and currently serving as a Scientist D and Joint Director at BIIS from November 2020 to the present. His research interests are development of vaccines, diagnostics, biomarkers, and recombinant proteins. He has 2 patents to his credit, more than 50 research papers published in international and national peer-reviewed journals, 4 book chapters, 10 GenBank submissions, and 22 abstracts for presentations in conferences and seminars. And he also serves as a reviewer for many peer-reviewed journals. So with this short introduction, I invite Dr. Amrutlal Patel to deliver his lecture. The floor is yours.
Okay. Thank you, doctor, for the very nice introduction. Now I will start today's topic on application of biotechnology to veterinary medicine. So briefly, I will introduce what is the biotechnology we can conceive nowadays and what is veterinary medicine, because many of the fellows will not be aware about veterinary medicine. So biotechnology is basically an area of biology that deals with the living processes or system or organism to manufacture the products or technology to improve the quality of life. And over the last couple of decades, we have come across the number of disciplines, almost every areas of biological sciences where biotechnology have been applied. Prominently, we can say that these are areas like animal biotechnology, medical biotechnology, plant biotechnology, industrial biotechnology, environmental biotechnology, and marine biotechnology. So almost it covers all areas of the life sciences. And GBRC is one of such place where we are working on all these areas. So if somebody want to pursue biotechnology career in any of this discipline, then GBRC is one of such place who can offer biotechnology in different disciplines. Veterinary medicine is basically a branch of medicine that deals with the prevention, control, diagnosis, and treatment of diseases, disorder, or injury in animals. And many of you must be aware that in veterinary medicine, we cover a range of different species like cow, buffalo, sheep, goat, horse, donkey, camel, elephant, pig, dog, cat, lion, tiger, other wild animals, as well as birds, including poultry. So from this number of different species, you can understand that these animals have different anatomy, different morphological features, different physiology, and even if you look at the medicinal aspects, they have different range of infections. Every species get different kind of infectious diseases as well as metabolic diseases. And if you look at the treatment, pharmacological point of view, then also the treatment differs from animals to animals because they have a different body weight, different physiology. So same drug, we will have to we will get the different response in different animals. So treating the animal in veterinary medicine is very tricky, and you have to remember a lot of different things for different animals. So somebody, if want to pursue the career in veterinary medicine, must be aware that it is going to be a very challenging task compared to human medicine. But there is one beneficial aspect is that we don't go in very much depth in veterinary medicine, like the advanced technology we use for human, like surgery, brain surgery, heart surgery, that kind of work we are not dealing in the animals. So in that way, it is relatively easy. So with this brief introduction, I will now focus on the biotechnology point of view. And in veterinary medicine, we have a number of applications for the biotechnology, including vaccines, development of vaccines, development of diagnostics, then developing technologies to enhance the animal production. Then there are assisted reproductive technologies like IVF and ICSI that are also used in the veterinary medicine. Then there is genomics, especially you can call metagenomics, which is an area which can provide potential applications in the veterinary medicine, especially for example, human metagenomics can provide you a number of different bacterial or fungal isolates or enzymes which can be used for the industrial production. Then there is a application for gene therapy, especially in the small animals. There are a number of people have developed the products for gene therapy. Then induced pluripotent stem cells, as many of you must be familiar with the stem cells, and iPSC is one of the stem cells, like embryonic stem cells. So the iPSC has also got a number of applications in the understanding as well as treating the diseases in animals, as well as for the application into the human medicine. Then there are applications like production of recombinant proteins or antibodies for the pharmaceutical purpose for treatment or diagnosis of animal diseases. Then there is a promising application of transgenic animal production where instead of producing the recombinant proteins in the fermenter using the bacteria or mammalian cells, you can directly produce the recombinant proteins in the milk or urine or serum of the transgenic animals. And that way you can produce the high value protein with a minimum cost and high yield. Then there is also a area where we can apply biotechnologies to introduce the disease resistance in animals, because many people have identified the disease resistance genes, and some breeds are resistant to particular infection or diseases compared to others. So by that way, by identifying genes from one animal or from one resources, we can integrate that gene into the another animal and make that animal resistance to the particular diseases. So first, we will talk about the vaccines. Everybody is very much familiar now because of the COVID-19. Whole country, in fact, entire world has been vaccinated for the COVID-19. So everybody is very much familiar with it. It is basically a substance which is used as an antigen. So antigen can be either inactivated whole organism or substance of the organism or nucleic acids or synthetic peptides which can be used as an antigen. And this antigen is basically used to stimulate the production of antibodies against particular pathogen. Not only antibodies, but also the cell-mediated immunity. Cell-mediated immunity means there are different cells like T cells are involved, helper T cell and cytotoxic T cells are involved in the eliminating the pathogen infected cells. So vaccines can also induce the antibody as well as the cell mediated immunities. So if you look at the important diseases in veterinary medicine, infectious diseases, these are listed here like brucellosis. This is a bacterial disease caused by Brucella abortus and melitensis in large animals, especially cow, buffalo, sheep, and goat. And Brucella is particularly zoonotic, even to human also, even any human beings which are coming in contact with the excreta of the animals infected with Brucella can also acquire the Brucella infection. Especially the veterinarians, veterinary doctors who are treating the animals, they are more prone to the acquisition of the Brucella infection from the animals. Then FMD is specifically viral disease, affects the cloven hoofed animals, especially cow, buffalo, sheep, and goat. Then tuberculosis, most of you are aware, it is a caused by the mycobacterium and it is a zoonotic. It can be transmitted from the animals to humans. Rabies also is zoonotic from animals to humans. Avian influenza normally occurs in poultry or chicken species that causes the zoonotic infection in human also. Infectious bursal disease and Newcastle disease are the poultry diseases, poultry viral diseases. Sheep pox and goat pox are the viral diseases in sheep and goat. Then also there is a pox related virus, lumpy skin disease, which affect the cattle, and there is recently outbreak in cattle is going on by lumpy skin disease virus. Then anthrax is also zoonotic disease caused by the bacteria. It is highly zoonotic and there is no effective treatment. Then leptospirosis, that is also a zoonotic disease, occurs in animals as well as in humans. Then canine distemper, babesiosis, trypanosomiasis, and thylaurisis affects in the animals. So these are some of the important diseases I have listed. I have not listed all, but major diseases which cause production loss in the animals and which are zoonotic into the humans. So for all these diseases, we can develop the vaccines to prevent the diseases. So to talk about the vaccines, traditionally we used to develop vaccine by conventional means, by growing the pathogenic organism and inactivating it by formaldehyde or beta propionolactone and making a killed vaccine. Or we used to propagate the virus in a non-natural host like chicken embryo, and for several passages like 50 odd passages, and then making it lost the virulence causing genes, or creating mutations in those virulence causing genes and making it attenuated, so it will be no longer pathogenic to the host. So like that, we used to make vaccines, but with the help of biotechnology, now we are developing a new generation vaccines based on the viral vectors based vaccine like Covishield, which everybody must have heard about. Covishield is a viral vector based vaccine. So similar viral vector based vaccines are also developed in the animal medicine. Then there are DNA vaccines like Zygo-D developed by Zydus Canada. These DNA vaccines are also developed for the animals. Then protein subunit vaccine, instead of DNA, you express the protein in the mammalian or bacterial host outside the animal target species. And those purified proteins you introduce as a vaccine. Then multiple epitope vaccines, where you can combine the epitope from the multiple antigens or multiple different pathogens to protect against the diseases, against a variety of diseases. Then mRNA-based vaccines are also, many of you have heard, like Moderna and Pfizer have developed for COVID, especially in the Western countries. People mostly have used the mRNA-based vaccines for the COVID. So if we look at the overview of vaccine development, basically when we get the new pathogen in the population, first of all we have to do is to identify which pathogen it is. And to do that, we have to sequence the genome of the pathogen. So after sequencing the genome of the pathogen, we have to identify that which proteins are structural proteins which makes the surface of the virus or bacteria, and those structural proteins whether they are able to induce antibody or not. And after identifying the neutralizing antibody inducing the inducing antigens, we have to clone those genes for the making recombinant vaccines, either DNA vaccine or subunity vaccine or vector vaccines. So this is the overall landscape of the vaccine development, starting from genome sequencing to the clinical trial. So basically, initially when we encounter pathogen, we sequence the genome and then identify the antigenic genes and then clone those genes in a suitable vector or express those proteins using suitable host, and then purify those vaccines or scale up the vaccine candidate if it is a virus. And then using this vaccine formulation, we have to initially screen in the mouse model or hamster model whether a vaccine are safe or not, whether the vaccines are effective or not, they are able to induce the antibody production or not, whether they are able to induce the cell-mediated immunity or not. Like that, all this preclinical testing we have to do in animal model. And once we identify that the vaccine is able to induce the neutralizing antibody and neutralizing antibody are able to counter the pathogen or neutralize the virus or protect against the infection, then we go for the human trial. And in human also, there are a number of different phases, phase 1, phase 2, phase 3, where we start from initially from 60 to 80 people, and then from second phase, you go for 100 to 1,000 people. And then in the third phase, you go into 10,000 to more than that number of people to study the safety as well as efficacy of the vaccine. So these are the number of different vaccine platforms used for the animal vaccine development. These are the examples of different animal vaccines like DNA vaccine, recombinant protein-based vaccine, chimeric protein-based vaccine, chimeric viral vaccines, as well as viral vector vaccines or RNA replicon-based vaccines. So here in the DNA vaccine, you just clone your gene of interest into the mammalian expression vector, and then that DNA itself you purify from the bacteria and check the quality of the purified DNA, whether it is free from endotoxin or not, and then directly inject into the animal, either intradermally or intramuscular. And that injected plasmid DNA gets transcribed in the cell after the injection and produces the recombinant protein which induces the antibody. So another approach is either instead of directly injecting the DNA, you can express the protein outside the host, that is in the mammalian cell or E. coli, and then that protein you purify, and that purified protein you inject into the animals. So that is a protein subunit vaccine. In that also you can combine multiple epitope or multiple protein from different pathogens or the same pathogens, and then produce the chimeric vaccine. Or there is a platform like chimeric viral vaccine where you express the antigenic protein on the viral coat proteins of the different vector and then use as a vaccine. Or you just clone the gene of interest into the genome of the viral vector that may not express the protein on the virus coat, but it will infect the cells after the injection into the animal, and after infection it will produce the antigen. So that is viral vector based vaccines. So these are number of different vaccines developed for the animals. This is a list of vaccines developed for the animals for different species like canine, feline, that is cat, then equine, horse, swine, that is pig, bovine, that is cow, buffalo, sheep, goat, then avian, that is poultry. So for different species, even for wildlife and rabbit also, vaccines are developed by different companies and against the different pathogens like canine distemper, feline leukemia, rabies, influenza, West Nile. So a number of different pathogens have been targeted using the different viral vector platforms like canary pox or bovine viral diarrhea or equine encephalitis virus or adenovirus based platforms. So these are number of vaccines which are commercially available in the market for the animals. Now, talking about the diagnostics, for diagnostics also we can apply the biotechnology based tools. Traditionally, what we used to do for diagnosis is if for infectious disease, we used to isolate, do bacterial isolation or viral isolation from the infected patient. Or for non-infectious disease, we used to do clinical hematology, blood parameters, and or microscopy, for example, for malaria. And then we used to diagnose the infection. But nowadays, with biotechnology-based approaches, we can go for molecular test, like detecting the antigen or antibody against particular pathogen, or even directly capturing the nucleic acids of the organism like DNA or RNA. In fact, for COVID, you must have heard about RT-PCR test. So that is based on the nucleic acid detection. Or many of you might have also seen that those strips were available where we used to put the nasal drop on the strip and we can see the bend, whether positive or not. That is based on the antigen detection. So these are some of the biotechnology based tests are available for detecting the diseases. So these are the list of diagnostic tests available for the animal veterinary market based on the PCR and real-time PCR. Also, number of tests are available from the IDVET. Then ELISA-based tests, which are based on the antigen antibody reaction, that are also available. Then rapid assays like lateral flow assays, that is strip-based assay that you have seen in the COVID. These are also available for the animal disease diagnostics. And there are latest technologies like next generation sequencing and microarrays can also be used for diagnosis. Next generation sequencing especially is used when you don't know, if you are not able to predict that which kind of infection it is based on the symptoms, then you can sequence the whole samples and identify what are the positive agents. Like this COVID, when there was a pandemic, the first detection of the pathogen was identified by next generation sequencing. Then afterward only we came to know that it was caused by the coronavirus. So NGS can also be used in cases where we are not able to detect the disease by other techniques. Then microarray is also one of the technique which can be used for detecting for diagnosis of copy number or aneuploidies, especially in the genetic diagnosis, in the prenatal disease diagnosis. You can identify you can identify the bacterial pathogens also with the diagnostic microarray, or you can identify the SNPs with the microarray techniques. So other topic we will move on is to development of technologies to enhance the animal production. So traditionally for animal production, people have started with this selective breeding. So by knowing that this animal is producing the higher milk compared to other, we were using those animals for the breeding. And likewise, we have improved the number of traits as well as the production amount of milk as well as meat by using the selective breeding. But nowadays we have a biotechnology based approaches like transgenic animals or knockout animals or genomic selection, which we can use for improving the production. So here is one example of the increased muscle production by knockout of myostatin gene. So this cow, Belgian blue cattle, is basically having the mutation in the myostatin gene, where you can see the double muscling phenotype, that muscle mass has doubled than normal. So myostatin, we can successfully knock out to increase the muscle mass in different species also, like sheep and goat, to increase the meat production. So in this direction, we at Anand Agriculture University worked for few years, working on the knockdown of myostatin gene, and we reached up to cell line level, and we knocked down myostatin gene and studied its effect on the gene expression program, and we developed the stable cell lines for myostatin knockdown, and published number of papers. So other approach is the genomic selection, where by which we can improve the production in the animal. So in genomic selection, what we have to do is first identify the markers through reference population. So if you have a high producing animals or elite animals, you screen those animals for the genotype or SNPs, and those animals SNP you link or associate with the production traits and derive some equations, and based on those equations associations, you can screen any animal right from birth to predict whether that animal is going to produce the higher milk production or higher meat production or not. So by doing the genomic selection, even at the embryo level also, we can do genotyping of animal to predict whether that animal is going to produce the higher production or not. Next topic is the assisted reproductive technologies. In that number of technologies people have used, first is the artificial insemination, where we administer the semen of animal, semen of bull directly into the uterus or vagina of the animal in the heat. So by artificial insemination, the target is to use the genetically superior male. When these males, we normally test by progeny testing and identify that this male is genetically superior and the progeny of those bull are producing high milk. Then semen of that bull we collect and make millions of doses and administer into the animals in the heat, during the heat. So by that we have improved milk production to a significant level. And the another technique is the multiple ovulation and embryo transfer that we will see in the next slide. The semen sexing, that is also one of the techniques. Like in the India, cow are considered as sacred. So if by natural breeding, what happens that 50% of the progeny we get is the female and 50% are male, and male are not productive. So male are going for slaughter. So ethically, it is not acceptable in India. So what we can do is we do semen sorting for the X and Y sperm. So X and Y sperm, there is difference in the DNA content. X sperm is having the higher DNA content than Y sperm. By binding with the DNA binding dye, we can identify that this X sperm has a higher fluorescent intensity than Y sperm. So based on the fluorescent intensity, we can apply the charge. So the higher intensity, we can apply positive charge, and lower intensity, we can apply negative charge and deflect those sperm into a different vial. So by that way, we can sort the X bearing spermatozoa into a different vial and use that as the insemination. So we can only get the female progeny. In 99%, 95% accuracy, we can get the female progeny. So by that way, we can enhance the production. Then there are techniques like in vitro fertilization and intracytoplasmic sperm injections that we are also applying in the animal medicine to increase the usage of the genetically superior female as well as male. Then embryo sexing can also be useful. Like in the semen sexing, some progenies are still born as a male. So if we do embryo sexing at the morula or blastocyst stage, we can identify that this embryo is going to develop into female only. So with that technique also, we can improve the production. Then there is a technique like somatic cell nuclear transfer that we will see in the subsequent slide. So this is the multiple ovulation and embryo transfer. This technique is particularly used for enhancing the use of genetically superior female. Like artificial insemination is used for enhancing the use of genetically superior male, whereas this MOET is used for using the genetically superior female. So normally during the life cycle, one female cow will produce 8 to 10 progeny. But by using this multiple ovulation and embryo transfer, that genetically superior cow, which is showing good milk production, we can use that cow to donate the embryo, like hundreds of embryo, and produce the hundreds of calf during the lifespan. So this technique particularly is used to enhance the production of genetically superior female. And this is somatic cell nuclear transfer, which is also called cloning. That many of you have heard that in 97, Ian Wilmot has produced the Dolly cloned sheep. That picture is given here. What they have done is from this Scottish blackface sheep, they have taken the embryo, the oocyte, and from this fin dorsative, they have taken mammary gland epithelial cell, and the nucleus of this mammary gland epithelial cell was transferred into the anucleated embryo of the fin dorsative. And after the fusion by electric pulse, this was grown into embryo and transferred into the surrogate mother. And you can see that progeny which is produced is a like fin dorsative, so from where nucleus is taken. So these techniques can also be used to enhance the use of genetically superior animals. Even from the adult somatic cell also we can use to produce the progeny instead of using the gametes. Now we will move on to another area, that is metagenomics. So metagenomics has tremendous application in the animal production. Like rumen metagenomics. Metagenomics is basically a sequencing the collection of genetic material or community as a whole instead of culturing. So normally what happens that if we culture the microbes or bacteria from a particular sample, we will be able to grow only 1% of the organism. 99% of the organism do not grow in a selective media which we use in the laboratory. So we don't know what communities are present in the sample. So to do that, we directly subject that sample to DNA isolation and sequencing. And after sequencing, we identify what are the different communities are present in that particular sample. Especially for rumen. Rumen, as many of you know, though it is a four stomach in the ruminant animals where there is fermentation is going on. So animal are eating grass, and grass is very difficult to digest. So if you so in the rumen there are bacteria and fungi and protozoa which collectively digest this plant material and provide the energy source to the animal. So these bacteria and protozoa harbors number of enzymes which are very much important or which are very much potent enzymes in degrading this cellulosic material. So these enzymes we can identify after sequencing of this metagenome. and then clone and express in the industrial scale. So these are the number of publications we have published from rumen metagenomics by working at Anand Agriculture University. Almost 42 publications over the span of 10 years. Then these are number of pet enzymes which we have isolated from the rumen of the animal. And these 4 papers were published for these enzymes isolated from the rumen. Another area is the gene therapy. Gene therapy is basically a technique to treat the diseases by introducing a gene. So this gene either you can use to replace the defective gene, or you can correct the defective gene, or you can introduce new gene in the diseased animal. So there are 2 approaches for gene therapy: ex vivo gene therapy and in vivo gene therapy. In vivo gene therapy, you can directly inject your medicine directly into the animal. Whereas in ex vivo, what you do is you take out the cell from the animal, and in vitro, in the laboratory, in those cells, you carry out the modifications by infection or transfection. And after modifying those cells, you replace back into the animal or human. That is ex vivo gene therapy. So there are a number of diseases in animal medicine, especially in canine medicine, which can be targeted for gene therapy, like retinal degeneration, retinitis pigmentosa, Leber's congenital amaurosis, X-linked retinitis pigmentosa, achromatopsia, hemophilia A and B, leukocyte adhesion deficiency. These are some of the examples of monogenic diseases which occurs in canine, which can be treated. And this is the list of different attempts people have made in canine to treat the monogenic diseases by gene therapy. Even it was started long back in 1993, where people have treated hemophilia B by correcting the blood factor 9 gene by retroviral vector. So likewise, for hemophilia A, muscular dystrophy, and other diseases, also people have made number of attempts, and these are the successful example of the gene therapy attempts in the canine medicine. Another area is the induced pluripotent stem cells. As many of you know that stem cells has the capacity to differentiate into the any type of cell. So in stem cells, there are 2 types. Embryonic stem cells and induced pluripotent stem cells. So embryonic stem cells you have to collect from the embryo. So embryonic stem cells have a limitations that for adult, if you need a stem cell, you cannot get the embryo again. So and also there are ethical limitations that even during the IVF, you cannot collect the stem cell and grow ethically. So induced pluripotent stem cells are basically you can differentiate from the somatic cell. So even using the adult somatic cell, you can differentiate into the pluripotent stem cells by providing the reprogramming factors. So these reprogramming factors are like OC4, SOX2, KLF, nano, this CMYC, all these reprogramming factors we can use to reprogram the somatic cell into the pluripotent stem cells. So these iPSC we can generate, people have generated for a number of species, including human, mice, canine, bovine, and there are standard protocols are available. So these iPSCs, based on the requirement, we can differentiate into either neurons or intestinal cell or hematocrit hematopoietic lineage cells or muscle cell, whatever is the requirement, and then we can transfer into the patient. So in veterinary medicine, iPSC is a promising platform for disease modeling, drug and toxicity testing, biomarker development, as well as cell-based therapy. Also, large animal represent a best model for man. Instead of doing testing in mice or rat, if you do testing of iPSC in large animal like canine or bovine or pig, they will represent more to the human for translating that technology into the human medicine. So even in addition to the translating into human medicine, you can directly develop product for the veterinary medicine, especially in the canine medicine or regenerative medicine. In the case of injuries in the sport animal like horse as well as canine, we can use the iPSCs for the treatment. So these are the different applications overall, like basic research to biodiversity preservation, biomarker development, cell-based therapy, disease modeling, transgenic animal generation, human organ generation in domestic animals, as well as gametes or embryo generation. So iPSC has a number of applications in all domains. This is one of the study we have carried out on developing the induced pluripotent stem cells at Anand Veterinary College. We worked on the canine induced pluripotent stem cells, and we were able to generate the iPSCs from canine fibroblasts, dermal fibroblasts. And we found that these canine iPSCs were in between mouse and human. So they have expressed some markers specific to mouse and some markers specific to humans. So there are representing the intermediate lineage of mouse and human iPSCs. So another application is the recombinant proteins, where we have potential applications in veterinary medicines. These recombinant proteins, you know that in the biopharmaceutical companies, they are making for the humans. Like FSH is made for the IVF. So same recombinant proteins we can also develop for the animals. In animals also we do IVF. So apart from FSH, we can also develop antibodies as well as other therapeutic proteins in the animals. So FSH and LH, as you know, that they are produced in the brain, pituitary gland situated below the brain. So from that, it releases the FSH and LH into the bloodstream, and those FSH and LH travels to the ovary of the uterus and acts on the ovary to mature and release the follicle as well as ova. So this FSH and LH has tremendous application in the IVF technology in the veterinary medicine. So basically what we have to do is to clone the alpha and beta subunit of the FSH gene and make the stable cell line based on the Chinese hamster ovary production cell line. And those cell line we have to scale up to the fermenter level to produce the FSH and LH to the production scale. And after producing, we have to do the structural and functional assays for the activity of the FSH and LH. And it takes almost 36 to 40 days to get the product. So all process up to QC test, it takes 7 weeks to release the product into the market. Another application is the single domain antibodies. Like other antibodies, there are single domain antibodies present especially in the camel. This single domain antibody is different from the conventional antibody in that in single domain antibodies, there is only one light chain. Whereas in the conventional antibody, there are 2 heavy chain and 2 light chain. Whereas in the single domain antibody, there is only 1 heavy chain only antibody. So this single heavy chain only antibody binds to the antigen and produces the effect. And it is very much small, like 15 kilodalton compared to 150 kilodalton size of the conventional antibody. And this single domain antibody can be used again for the treatment of viral diseases like bird flu or Crimean Congo fever, or even this antibody can be used for development of diagnostics. And we can develop this antibody by fast display or bacterial display technique and express in the bacteria or expression system. So another application of this biotechnology is the transgenic bioreactors. As I have mentioned in the introduction, that we can produce the recombinant protein in the milk or urine or serum of the animal instead of doing it in the fermenter. So what we have to do is to clone the therapeutic gene into the transfer into the embryo, and from embryo we have after integration we have to transfer into the animal and produce the progeny and screen the progeny for the presence of the transgene and expression of the protein. So by this technique, we can produce the therapeutic protein into the milk or serum or urine of the animals. And these are number of successful example of producing the human therapeutic proteins into the milk of transgenic mouse and rabbit using the milk protein gene promoter, and then they are commercially viable, and you can get the cost-effective therapeutic proteins in the animal milk. Another application is the disease resistance. So traditionally, by selective breeding, initially from wild animals, we have domesticated the animals, and through selective breeding, we improved the milk production. But latest, there is genomic selection, which is used to increase the productivity traits by genotyping the multiple SNPs and associating with the phenotype. But the limitation of this technology is, if you want to introduce a new character, then by breeding it is not possible. So if you want to introduce a new character, you can directly introduce new gene by transgenesis, or you can directly knock out the disease causing gene by gene knockout or CRISPR-Cas9. This is the latest technology to introduce the disease resistance. So here is one example where people have done knock-in animal with introduction of SP110 nuclear body protein in the cattle. This cattle have been found to be resistant to the tuberculosis. So this by introducing this SP110 protein by talent-mediated transgenesis, introduction of the SP110 gene, this animal was found to have the stimulation of apoptotic pathway instead of necrosis pathway from the in the mycobacterium infected cell, and that resulted in the immunity against the mycobacterium and resistance to the disease. So this is one of the examples. So likewise, we can identify more genes to treat to introduce the disease resistance in animals. So overall, these are the summary of different applications of biotechnology in veterinary medicines. There can be much more applications apart from what I have discussed today. So with this, if there are any questions, I will be happy to answer.
Thank you so much, Doctor. The session is open for discussion.
Sir? Sir,
Yes.
I had 2 questions. One question is, you were mentioning about chimeric vaccines. So, vaccines from based on subunits of different viruses, if I'm right. So how do you ensure that they're folded correctly so that the correct antigenic portion is accessible to produce an immune response?
Basically, folding is a issue in the when you are expressing protein in vitro. So if you are expressing in the natural host, like mammalian cells, if you are using human cell lines or animal cell lines, then the fold likeliness of the folding, correct folding is there. But if you are doing expression into the E. coli, then there is a possibility that protein may not fold properly. But at the end, what is important is the neutralizing antibody. When you do a preclinical trial in the animals, you get a antibody against the target antigen, and that antibody you test for the virus neutralization. And if your antibody in the immunized animal, or if sera from the immunized animal is able to neutralize the virus, that serves your purpose. So what we want is whatever antigen we are using in a vaccine, that should be able to induce the neutralizing antibody, or that should be able to protect against the infection. So we are doing a series of tests like challenge infection with a virus. So whether it is protecting or not. So that is the ultimate proof that vaccine is working or not.
Okay, sir. The other question I had was when you were talking about iPSCs and how they could be used in basic research. Is it possible to use it to test wound healing? If so, could you explain a little more about that?
Possible to use for wound healing.
To study wound and healing.
Yes, definitely. For wound healing, there is a potential applications for the stem cells. So we can directly use the stem cells for the in the application of wound healing. But there is one concern is the teratoma formation. So still this technology has not gone to the clinic. Especially when we are integrating the this transcription factors like OSKM viral vector. Then for making the iPSCs, those iPSCs, there are chances that they are likely to cause the teratoma. So for wound healing, potential application is there, but still we have to refine the technologies so that it cannot cause the teratoma.
So just to clarify, I wanted to ask if we could use this as a model to study a wound and test a therapy for wound healing. Like, so even then does this apply? The chances of forming a teratoma?
So definitely we can use is a model for wound healing as well as therapy for wound. But again, we have to eliminate the concern of teratoma formation. So for that, iPSCs which are generated, we have to characterize in a way that they are not they are not causing the teratoma formation.
Okay, so are there any papers that I could read related to that? Like, is there any work? I will go look for it.
That I will have to find out. If you are particularly looking for the iPSCs in wound healing, then you can share your ID in the chat box. I will find out the papers and send it to you.
Sure. Thank you so much.
Okay.
Doctor, we have a question in chat box.
The question is pretty long. Actually, the question is such that why we see outbreak in the animals which are domesticated and not which are wild. But that question, I think it is not absolutely true, because 2 years back there was outbreak of canine distemper virus in lion population in Gir forest. And last year also you might have heard that there was yeah, in avian influenza outbreak in many migratory birds, and many bird species have died in the just 1 year back. So it is not like the outbreak we see in the domestic animals only, but we are noticing outbreak better if it occurs in the domestic animal, because we are closely living nearby them. Otherwise, outbreak also happens in the wild animals as well as birds.
Any other questions? One more question in the chat box.
So another question is how xenotransplant are prepared. Recent example of xenograft porcine kidney used in the human patient. So basically, there are human leukocyte antigens which are creating the problems in the rejection of the xenotransplant. So in this, from the animal, when we are making xenotransplant, we have to knock out those leukocyte antigens which can be detected by the host. So by making the transgenic animal, we can make it more humanized organs for the xenotransplantation.
Any other questions? Anyone else? If no more questions, then I would like to thank Dr. Amrutal Patel for joining us today and educating the students about the vaccines and development. Thank you so much, Doctor, for joining us today.
Thank you, sir. Thank you for providing the opportunity. Thank you.
Thank you.